All right, everyone, we are live. A very good afternoon and warm welcome to Shri Chaitanya. This is your NEET 10, a game changer series, and today we are here with day 6 with the botany session. And in every session, we are doing 25 most important questions from 25 different topics, and these topics have been picked up from the previous year questions analysis. So basically, we have picked up 10-year NEET PYQs and finalized certain concepts from which we are repeatedly seeing the questions. Okay, so from those particular concepts we have made 25 different concepts per session. All right, everybody, hellos and hi. So first thing first, please do hit the like button and share it with all your friends, and then let's get started with our super amazing class of botany with 25 different concepts for this particular session. Hello Siddhant Yadav, hello Yoga, hello Janavi, hello Sai Ram, hello and good afternoons. Come on everybody, quickly join, quickly. Do hit the like button and share it with all your friends. See, today is 30th of April. We are left with just 4 days, and on 5th of May, we have our D-Day. We have our final NEET exam 2024. Yes, with lots of hellos and highs, let's get started. This is your NEET 10, a game changer series, which claims that you're going to score 650 plus marks by this particular series, because in every session you are going to solve 25 amazing questions from 25 different concepts. All right, and this is the schedule for your sessions from 12 to 5:30. The timing for 3 to 4 is of botany class, and I am Beena Yadav. I will be catering your botany sessions. After that, you will be having class of chemistry from 4:30 to 5:30. All right, let's get directly jump into the questions, everybody. Hello, Black Sea. Hellos. So let's get started, bacho. This is your first question. First question, everybody, on your screen. First question, come on, kids. Phosphorylation of glucose during glycolysis is catalyzed by. The very first question on your screen, kids. Phosphorylation of glucose molecule. This question is basically from respiration chapter, and specifically from respiration we have the first pathway, which is glycolysis. Okay, so this question has been picked up from glycolysis, which is a 10-step pathway, and I can see answers. Study, work hard has given the answer. Amrita has given the answer. Avish has given the answer. Sai Ram, hello, please answer everybody. Yes. The question is, phosphorylation of glucose during glycolysis is catalyzed by. So first of all, if I speak about glycolysis, lysis simply means splitting. Okay, lysis is splitting, and glyco means glucose. So basically, there will be splitting of glucose molecule. All right. The very first step in glycolysis is we have glucose molecule acting as a substrate. Then basically, in this glucose molecule, my 1 ATP will be broken down into ADP plus PI. So please pay attention, kids. 1 ATP has been utilized here, okay? And this phosphate which is there, that will be given to the glucose molecule. Thereby, it will form glucose 6-phosphate. This is the first step of glycolysis. Since we are adding phosphate, that is why this reaction is called as phosphorylation reaction. Okay, and this reaction will be catalyzed by an enzyme called as hexokinase. Now, why kinase? Kinase is a group of enzyme that acts whenever we want to add phosphate or remove phosphate. In that case, the group of enzyme that has been used is kinase. And hexo simply means 6. My glucose molecule is 6 carbon compound. Okay, my glucose 6-phosphate, again 6 carbon compound. So basically the substrate is 6 carbon compound. That is the reason hexokinase will be used. Hexokinase is a general enzyme, we can say. More specifically for this particular step, we can also use glucokinase. Okay, hexokinase is general in a way. Glucose is 6 carbon compound, fructose is also 6, galactose is also 6. So for all these molecules, be it glucose, be it galactose, be it fructose, my hexokinase will be utilized. But this glucokinase, this is specific, this is exclusive for glucose molecule only. Okay. Yes, so that is why answer will be my option number C, which is hexokinase. Yes, can I see plus 4 in this question? PTK, hello, hello, hello. Hello, Neer, good afternoon. Can I see plus 4 in this question, kids? Can I see plus 4s in this question? Yes, yes, bachas, plus 4. Great. Let's move to the second question then. Next question, kids. Next question. This is assertion reason type of question, and you know the drill. Whenever we are confused between option 1 and 2, we just need to hello Ghana, hello, hello. We just need to follow a simple step, which is just read out first statement, which is assertion, put a big question mark and try to find out answer of this question. If we get the answer in reason statement, in that case, answer is option number 1, otherwise it is option number 2. Yes, come on, try to solve this question. Everybody. Everybody. Try to solve the question. Assertion statement says in some leguminous plants, the leaf base is swollen. Okay. Specifically in the case of leguminous plants, the leaf base is swollen. The reason says swollen leaf base is called as pulvinous. Pulvinous. So why the leaf base is swollen? If you get the answer, then option 1. Yes, Neer and Priya Sharma has answered. Sudhakar has also answered. Siddhant has answered. Amrita has answered. Great, kids. It is your option number 1. Of course, the swollen leaf base is called as pulvinous, and your leguminous plant, in case of leguminous plant, the leaf base is your swollen. Okay, so it is your option number 1. Yes, yes, Priya Sharma. Yes, PTK. Yes, Ghana. Avinash, Sudhakar, Siddhant, Amrita. All right. So answer is option number 1, plus 4 and minus 1 in this question, and moving on to the next question. Next question. We have 4 to 5 AR type of question because you people were asking for AR type of questions. So I have kept 4 to 5 questions back to back. We are AR type of question. Okay. Okay. So the first statement assertion is Selaginella and Salvinia are homosporous, and the reason says similar kind of spores are produced in Selaginella and Salvinia. Both the statements are correct or incorrect. Amrita has already answered. Rest of the kids, come on. Come on, come on. Rest of the kids, come on, come on. Selaginella and Salvinia. Siddhant Yadav, great. Amrita. Neer, think about it one more time, I would say. Yes. If I speak about pteridophytes, okay? The students who are giving answer as option number 1, please, please, please pay attention. If I speak about pteridophytes, my dear kids, in that case, see, up to bryophyte, the condition is homosporous. Specifically in the case of pteridophyte, your heterosporous condition starts coming, and we have certain exception like Selaginella and Salvinia. These two are basically heterosporous. Okay, they have 2 different kind of spores. So presence of this heterosporous condition basically marks that seed habit development is going to be the start. So in pteridophyte, seed has not been formed, but seed habit development has been started. Okay, so basically your female gametophyte will be retained on parent sporophyte, and basically your seed habit development has been started from pteridophyte onwards. Specifically, the plants which have heterosporous condition, in those plants only we're gonna get this condition. And examples are your Selaginella and Salvinia. So these are not homosporous. These are basically your heterosporous. All right. And heterosporous simply means the plant having presence of 2 different kind of spores. So they have different kinds of spore. Okay. So since the first statement is incorrect, that is why answer is option number D. This is incorrect. This is also incorrect statement. Okay, that's why answer is option number D. Is this clear, Amrita? Is this clear, Siddhant? Is this clear, Priya? Shiny? Ghana? Yes, not a problem, Neer. Okay, moving on to the next question. This is, I would say, very simple question, very simple and straightforward question. The embryo sac of an angiosperm is made up of how many cells? The embryo sac is made up of, very simple question, kids. It is very simple question. Embryo sac simply is, let me draw it here. This is your typical embryo sac in which we are able to see over here we have three, these are three antipodal cells. Antipodal cells. Here we have 1 and 2 synergids, and the central is your egg cell. So this is my egg, and these two, these ones are my synergids, okay? And in the center, from this part to this part, this is secondary nucleus, and these are my polar nuclei. Polar nuclei, okay? Let's count the number of the cells and the nucleus, my dear kids. If I speak about number of cells, I am having 1, 2, 3, 4, 5, 6, 7. So total we have 7 cells. It is 7 celled. What about nucleus? We have 1, 2, 3 nucleus. In the central cell we have 2 nucleus, 3 and 2, 5, 6, 7, 8. So it is 7 celled and 8 nucleate structure. Right? It is 7 celled and 8 nucleate structure. Very easy peasy question and commonly asked in our PYQ. Okay? So my embryo sac is having in total 1, 2, 3, 4, 5, 6, 7. 7 cells and 8 nucleus. 1, 2, 3, 4, 5, 6, 7, 8. That is why answer will be option number B, which is 7 celled and 8 nucleate. Is this correct, Amrita? Yes, Grace. Yes, Avinash. Siddhant Yadav. Priya, 7 celled, 8 nucleate. Hello, Karun. Dr. Muhammad, PTK, Priya Sharma, Shiny and Shalu. I hope it is clear now, okay? And do hit the like button also for these amazing questions. Come on, everybody. Yes, okay. Hello, Arjun Reddy. Moving on to the next question then. Next question, kids. If the diploid number of a flowering plant is 36, what would be the chromosome number in its endosperm? Okay? So, we have diploid number which is 2n. That is 36. What would be the chromosome number in its endosperm? In its endosperm, the chromosome number has been asked. Okay? Yes, yes. Simple mathematical question. The value of 2n has been given as 36. Endosperm is a product of primary endosperm nucleus. Primary endosperm nucleus is product of triple fusion, wherein we have 1 male gamete and 2 polar nuclei. Okay, so in total we have 3 nucleus which got fused, which means the primary endosperm nucleus is 3n. Okay, so endosperm will also be 3n. All right, the value of 2n is 36. The value of 3n will be. The value of 3n will be. Come on, kids. It cannot be option B. Value of 2n is 36. Okay, let's just understand it this way. The value of 2n is 36, so value of n will be 18. Now tell me what is the value of 3n, my dear kids? It will be 54. Is this clear? In the question, value of 2n has been given, which is 36. Clear now, Dr. Muhammad, PTK, Priya Sharma, Shiny and Shalu, Siddhant Yadav, Grace, Amrita, Parmesh, yes? Is this clear now? Yes? Answer is option number C, which is 54. Clear, everybody, Ghana. Okay, moving on to the next question then. Next question, kids. It is again assertion reason type of question. Your assertion says it was such a beautiful question, no? Why endosperm is 3n? Arjun Reddy. Okay, let me take up this doubt. Arjun Reddy is asking why primary endosperm nucleus is 3n. Okay, if I speak about angiosperms, Arjun, so basically we have presence of 2 male gamete. Your 1 male gamete basically combines with the egg cell. Okay, your 1 male gamete combines with the egg. Male gamete is haploid, right? Egg is also haploid. So the product, my dear kid, would be your zygote. Zygote formation is there, which is n plus n comes out to be 2n, right? Now this zygote will later on form embryo. It will form embryo. That will also be 2n. Clear? Okay. Now what about the second male gamete? The second male gamete that we do have, second male gamete, this will combine with polar nuclei, the central polar nuclei which are there. All right, and polar nuclei are your N plus N, the central cell. Remember, just before 5 minutes I made diagram of embryo sac. In the central one we had 2 nuclei. Okay, so the second male gamete is going to combine with this polar nuclei. The product of this fusion is your primary endosperm nucleus. Okay, and this process is called as triple fusion. This process is called as syngamy. Right? Now, in case of primary endosperm nucleus, we have N plus N plus N, which is 3N. That is why primary endosperm ploidy will be 3N. This will later on will form endosperm. Endosperm. Since we have formation of embryo, which is 2N, and endosperm, which is 3N, we call it double fertilization because we have syngamy as well as triple fusion. Okay, that is unique property of angiosperms. Is this clear now why it is 3N? Yes, I hope it is clear now. Right, Arjun, ready? Why it is 3N? Yes. So for such an amazing question, do hit the like button. Come on, kids. Okay, next question, which is assertion and reason type of question. Assertion says archaebacteria are able to survive in harsh habitat. Okay, now you need to find the solution. Why harsh habitat? Because the presence of peptidoglycan in cell wall helps archaebacteria to survive in extreme conditions. Archaebacteria are the ones which survives, which thrives in extreme conditions, extremities. Okay, it is because of the presence of peptidoglycan in their cell wall. Is this correct or not? Hello, Ramcharan. Is this correct or not, kids? Come on. Is this correct or not? Yes, yes, I am getting the answers. See, there is a different cell wall structure. That is correct. There is different cell wall structure, but peptidoglycan is found in cell wall of bacteria, not in the cell wall of archaebacteria. In case of true bacteria, you're going to find peptidoglycan layer, basis of which we have 2 different type of bacteria, gram-positive and gram-negative bacteria. That is purely based upon whether they are giving positive result with respect to gram staining or not. Okay, so this reason statement is incorrect, but this statement is perfectly correct. That is why answer is option number C. Assertion is true, but reason is false. All right, everybody. So mark a +4 and -1 in this question, and moving on to the next question. Come on. Next one, fusion of 2 motile gametes which are dissimilar in size is termed as. Remember in the previous session, yesterday's session, I explained you about isogamy, anisogamy and oogamy in detail. Okay, those of you who are regularly attending the classes, the students who are consistent in their efforts would resonate with the fact that in yesterday's class I have discussed differences between isogamy, anisogamy and oogamy. Now the question is fusion of 2 motile gametes, gamete and motile, which are dissimilar in size. The size is different. Okay. Yes, yes, Rajya Lakshmi. The size is different in case of anisogamy. See, if I speak about isogamy, iso simply means similar, which means the gamete which are in the question has to be similar in terms of size, shape and everything. Okay, if the gametes are motile, both the gametes should be motile. If they are non-motile, both the gametes should be non-motile. Okay, if I speak about anisogamy, aniso means different, which means they are different in their sizes, different in their motility, everything would be different. If they are following oogamy, Then oogamy specifically is when we have male gamete, which is having small size and preferably motile, and the female gamete is having large size and it is static. So when we have this much of difference between the size of male and female gamete, that would be oogamy. So if you want to remember oogamy, typically take the example of human beings, okay? In case of human beings, trillions of sperms are there, and the size of the sperm is very tiny with respect to egg, okay? So egg is large and static at one place, but your sperms are, you know, a small size and they are motile, okay? Yes, everybody? Yes, the answer is option number C, since we have dissimilar size is given in the question, okay? Yes, +4 everybody. +4, and moving on to the next question. Association between mycobiont and phycobiont are found in mycobiont. Myco means fungal component. Phyco means algal component. Algal component. Yes. Yes, kids. Come on, come on. They are found in the case of lichens. What are lichens? Grace, Amrita, Neer. Lichens are basically symbiotic association of your algae and fungi. If your algae and fungi living together symbiotically in mutualism, then we call it lichens, okay? The algal component which is there, that is called as phycobiont. The fungal component is called as mycobiont, okay? So they are basically in a symbiotic association called as lichens, okay? Answer will be option number C, which is lichens. Clear everybody? +4, and next question, come on. Next question on your screen, assertion reason type of question. Assertion says gymnosperms do not produce fruit. They do not produce fruit, okay? Reason says why they do not produce fruit. Ovule of gymnosperms are enclosed within the ovaries. That is the reason they do not produce fruits. Come on. Good afternoon, Avinash. Good afternoon. Come quickly, bache. Yes? What is the reason why gymnosperms are not able to produce fruits? Because the ovule of gymnosperms are enclosed within the ovary. Is this the reason why they have naked seed? Yes or no? Priya Sharma, Amrita, Boya. Yes, yes. Assertion is true, but reason is false. Okay? Gymnosperms, gymno means the naked, sperms means seed, okay? The plant which have naked seeds. So forget about the fruits, okay? The seeds are also not enclosed inside any structure. They are open, okay? That is why we call them naked seeds. If I compare it with the fruits of angiosperms, in angiosperms we have flowers, we have fruits, and seeds are enclosed inside the fruits, all right? Yes, so first statement is correct, but reason is incorrect. Okay, kids, moving on to the next question then. Contagium vivum fluidium, very famous line that was proposed by. Contagium vivum fluidium simply means there is infectious living fluid. Okay? Infectious living fluid. This is proposed by. Great answers proposed by Bizring. Great answers proposed by Bizring. Perfect, kids. Perfect. Answer is option number B. Moving on to the next question, kids. Next question on your screen. Fill in the blanks are granular structures first observed under electron microscope as dense particle by which scientist? Fill in the blanks are granular structure and observed by, okay, let me give you one hint. By the name of that scientist, we also call the molecule by the name of the scientist. Adding particle. Great. It is ribosomes, and ribosomes are also called as pallid particle, simply to honor the scientists who have discovered them. Okay, so ribosomes are basically your Ribosomes are granular structure first observed under electron microscope as dense particle by Pallid. All right. So answer is option number 1. Option number 1. Perfect answer. Shiny and Shalu, Karun, Priya, Gana, Avinash. Perfect answer, kids. Moving on to the next question. +4 for this one. And let's move to the next question. Come on. Assertion reason type of question. Very interesting. Assertion statement says Avicennia has nematophores. Avicennia has nematophores. Put a big question mark. Try to find out answer of this question. Why nematophores are there? Or what are the nematophores? Nematophores help the plants to get oxygen for respiration. We call them respiratory roots. Amrita has answered, Yogesh has answered, Grace has answered. Rest of the kids, come on. PTK has answered. Rest of the kids, come on, answer. Be super quick. Gana has answered. Shiny and Shalu Diaries has answered. Great. Both the statements are correct, but this is not correct explanation because Avicennia has nematophores, okay? Nematophores are my respiratory roots. Okay, this is entirely different statement that respiratory roots help the plant to get oxygen for respiration. See, if I compare normal roots with respiratory roots, my normal roots are positively geotropic, meaning they are inclined towards the soil, inclined towards the earth. And they are negatively phototropic, meaning they are basically going away from the sunlight. But if I speak about nematophores or the respiratory roots, they are just opposite of this one. They are positively phototropic and negatively geotropic, which means they are coming out of the soil just for the purpose of respiration. The common examples of respiratory roots are your Rhizophora, Avicennia and Sonartia. These are the 3 plants which commonly have respiratory roots or nematophores. Basically, on nematophores, which are my respiratory roots, we have pore-like structures, and those pore-like structures are called as nematothodes. Okay, nematothodes are basically the pores responsible for respiration. All right, so the correct answer will be option number B. Both the statements are correct, but this reason is not the correct explanation of assertion. That is why answer will be option number B. Are we clear in this question? Everybody, are we clear in this question, kids? Are we clear? If we are clear in this question and you know, we are liking these kind of questions, then please encourage us by liking, hitting the like button, liking the session and sharing it with all your friends. Yes? Okay? Okay? Perfect? Okay. Next question then. Come on. It is match the column type of question, kids. We have column 1 here and column 2. Okay? Column 1, we have chloroplast, chromoplast and leucoplast. In column 2, we have got colorless plastid, yellow, orange or red color plastid and green plastid. And green. Yes, kids. Green. Chloro has, this word has been taken up from chlorophyll. The characteristic color which my chlorophyll gonna give is green color. Right? Great answers, Parmesh, Amrita, Avinash, Gana, Shiny, Neer. Very nice. Beautiful answers. Great answer. Correct answer is option number B. Almost everybody has done it right. Rebecca, Rakshan, great. Grace, great. Okay. Answer is option number B. See, chloroplast. Basically, it is your green plastid, having presence of chlorophyll molecule, and chlorophyll molecule has characteristic green color. Okay, so it is green plastid. Then we have chromoplast. Chromo word has been taken up from colors. So we have lot many colors in chromoplast, which means we can have yellow, orange or the red color plastid. Then we have leucoplast. It is colorless plastid, basically responsible for storage of various things like amyloplast, elioplast, alluroplast. Okay, Karun? Yes? So answer will be option number B. Plus for everybody in this question. I can see 100% students have given the correct answer. +4 in this question. And moving on to the next one. Come on. Next one. I guess I gave you answer of this question already. Yes, I already gave answer of this question. Amyloplast, elaioplast and aleuroplast belong to category of plastid. Which plastid? Yes. Amyloplast, elaioplast, aleuroplast, they belong to category of plastid. Which category? Do they belong to chloroplast, chromoplast or leucoplast? Come on, kids. Yes, ma'am. Yes, ma'am. It is option. Oh my goodness. Why option D, my dear kids? Just now, before 2 minutes, you can rewind the session. I discussed that in case of leucoplast, which is colorless plastid, basically responsible for storage of molecule. We have amyloplast for starch. We have elaioplast. We have aleuroplast for protein, elaioplast for oils and fat. So basically, depending upon which molecule they are going to store, we have named them differently, and all of them belong to leucoplast. Okay, chloroplast has got chlorophyll molecule. It has got chlorophyll molecule. Okay, chlorophyll. A, B are there, right? Then we have chromoplast, the plastid having colors, chromo, okay? So if, let's say, your leaf are little different in color, sometimes orangish color is there, sometimes little yellowish color is there. If my flower is of different color, that color is added by your chromoplast. By chromoplast, okay? Yes, and leucoplast is responsible for storage of different substances. All right, everybody, so answer will be option number C, which is leucoplast. I hope it is clear now. Not a problem if you made silly mistake, not a problem. Now I hope it is clear to all my bachas out there. Is it clear now? Because our exam will be on 5th of May. Today is not an exam. We can still learn so many things. We can unlearn certain things. We can relearn certain things. Yes, Karun? Yes, yes, kids. Got it, everybody? Yes, okay, perfect. Next question then. Next question. Lipids are arranged within the membrane with. Lipids are arranged within the membrane. If I speak about membrane, which is made up of phospholipid, we have bi-layer of phospholipids. Okay, so lipids are arranged within the membrane with. You might have seen this diagram wherein these are my lipids, okay, and this is my transmembrane protein, I can say. Again, my lipids are there. Have you seen this diagram of plasma membrane? Have you seen this diagram of plasma membrane, my dear kids? Lipids are arranged within the membrane. Head are towards outer side. This is your head part. This is your tail part. So as we all can see, my head is on the outer side, my tail is on the inner side. My head is on the outer side, my tail is on the inner side. Okay, so the correct answer is head will be towards outer side and tail are towards the inner side. Is this clear? Yes, Priya Sharma. Yes, Avinash, Neer, Ribak, Amrita, Grace. Is this clear now? This is clear? Okay, perfect. Perfect, everybody. Next question then. According to the modern concept, cell membrane is the diagram which I have made here. This is your plasma membrane, or we can say cell membrane. So the nature of cell membrane is it solid? Is it fluid? Is it quasi-fluid? We can say semi-fluid in nature. Or is it solidified sheath? Come on. Singer and Nicholson, sir, gave this fluid mosaic model of plasma membrane. So according to this fluid mosaic model, the arrangement of lipids and protein is there. So we call that quasi-fluid, or we call it solid, we call it fluid, or we call it solidified sheath. Come on. I want 100% correct answer in this question. Yes, Shalu? Shailu? I want 100% correct answer. Arjun ready? Ribak, Gana, Neer, Karun? Avinash, perfect everybody. Amrita, perfect. It is option number B, which is quasi-fluid, semi-fluid nature. Okay, this was given by Singer and Nicholson. This is effort of two scientists, Singer and Nicholson. They gave this model which says that we have this bilayer of lipids and proteins are embedded inside the layer, as well as we have some extrinsic proteins which are on the outer part of the layer also. Okay, so yes. So they basically told that the nature of this bilayer is basically quasi-fluid, which is semi-fluid in nature, completely not fluid. All right, so correct answer has to be your option number B, which is quasi-fluid. Perfect everybody. Let's move to the next question. Next one on your screen. Lysosomes are the reservoirs, storehouse of what? Lysosome. If I speak about endomembrane system, so endomembrane system in which we have different organelles connected so that they can package and modify certain materials. So those different organelles are basically number 1 endoplasmic reticulum, number 2 Golgi apparatus, number 3 lysosome, number 4 is vacuole. Okay, the question is from endomembrane system only. It says lysosomes are the reservoir for what? Why we call lysosomes as suicidal bags? What property they do have? Why they are suicidal bags? Great answers. Neer, Ribak, Karun, Priya Sharma, Kailash. Okay. Arjun, Shailu, come on, Neer, it is your hydrolytic enzyme. We have different kind of hydrolytic enzyme. We have lipases, we have proteases, we have all sort of enzymes. Basically, lysosomes are called as suicidal bags. If, let's say, we have any unwanted molecule which is there, so basically we will keep that molecule inside the lysosome, and it is responsibility of lysosome to digest that molecule. Okay? To digest that molecule. So answer is hydrolytic enzymes. We have many hydrolytic enzymes in case of lysosomes. Correct everybody? Yes. With +4, let's move to the next question then. In next question, we have column 1 and the column 2. Okay, so basically we have different type of cells, and here we have the function of the cells. Okay, the first one is bulliform cells. The first one is bulliform cells. Then we have guard cells, we have lenticels, we have subsidiary cells. Okay, bulliform cells. Come on, you try your level best, then I'll explain. Bulliform cells regulate opening and closing of a stomata. No, cannot be a function of bulliform cells. Aerating pores in the bark of the plant. It is my lenticels. Rolling in and out of the leaves. Yes, of course. My bulliform cells has got property to roll in and out so that the exposed surface area can be minimized. If the exposed surface area can be minimized, the loss of transpiration could be minimized. Specifically found in the case of grasses. I'm getting the answers. Very good, kids. Very good. So bulliform cells are basically helping in rolling in and out. Guard cells. Guard cells can be your dumbbell shape, can be your bean shape. They are basically regulating opening and closing of the stomata. Okay. If I speak about your dicots, we're going to get guard cells which are bean shaped. If it is monocot, the shape of the guard cells will be dumbbell shape. Okay. So guard cells help to regulate opening and closing of the stomata. What are lenticels? When we see secondary growth, in that case, we have seen when there are many complementary cells which are there. So basically the force that these complementary cells are adding on the outer layer of epidermis, due to which my epidermis will burst. It will permanently burst, and that will create a permanent open pore. This permanent open pore which has been created is called as lenticels. Now this lenticel is responsible for exchange of gases. Okay, responsible for exchange of gases, right? So lenticel are aerating pore in the bark of the plant. Now, there is difference between lenticels and stomata. We can regulate opening and closing of stomata, but we cannot regulate opening and closing of lenticels. Reason being, lenticels are permanently open pore. These are lens shape, but stomata, by making turgid conditions in the case of guard cells, by making flaccid conditions, we can basically maintain opening and closing of stomata. Okay. Next, we have subsidiary cells. These are the accessory cells. If I speak about stomatal apparatus, these are accessory cells. So my correct answer will be option number B. We can see A is 3, B is 1, C is 2, and D is my 4. So my correct answer will be option number 1. Okay. Great, everybody. Moving on to the next question then. Next question, kids, on your screen. Yes. Next question, assertion reason type of question. Assertion says the alternate type of phyllotaxy is the arrangement of leaves in which a single leaf arises at each node in alternate manner. Single leaf is there in alternate manner. The alternate type of phyllotaxy is seen in China rose and mustard. Okay, we can have alternate, we can have opposite, we can have world type of phyllotaxy. If I speak about alternate, supposing this is my plant, okay. So in case of alternate, one leaf is on this side, second will be on this side, third on this side, fourth in this side. In such a way, your one node will give rise to just one leaf. If it is opposite type, in that case, my one node is having two leaves. One is on this side, one is on this side. Okay, in case of opposite, your one node will give rise to two leaves. In case of world type of phyllotaxy, my one node will give rise to more than two leaves. Okay? Yes, I'm getting the answers. Great, kids, great. So, both the statements are correct, but this is not the correct explanation. Examples given are, of course, correct. China rose and mustard will be having alternate type of phyllotaxy. But if this is not giving the reason that why alternate type is present, okay? So this statement is correct, and this is not correct explanation. That is why answer is option number 2. All right, kids, with +4, let's move to the next question. To get a carpet like grass, lawns are mowed regularly. This is done to what? We want a carpet like grass, okay? You might have seen the gardener. The gardener is having this very big machine, and regularly it is moving that machine on the grasses. Yes, why is that so? You might have seen that. Why is that so? Why the lawns are mowed regularly? What is the scientific reason behind this? What is the scientific reason? Great answers, great answers. Come on, come on. You can see the screen. I'll be on side. Come on, kids. Come on, come on, come on, kids. Yes, yes. Basically, they are removing the axillary bud so that they can accelerate the growth of terminal bud. Okay? Yes, Amrita, great answer. Yes, Parmesh Boya, great answer. Priya Sharma, great answer. Yes, Karun, great answer. Okay? So answer is option number D. Both the statements are correct. All right. Next question. It is a last question for today's session. Assertion reason type of question. Assertion says, this symbol, it is the symbol for inferior ovary. Okay. Your reason says adhesion is indicated by enclosing the figure within the bracket. Yes. If your hyphen is below the G, is this inferior or superior? And is this inferior or superior? And what do I mean by superior or inferior ovary for that matter? Come on. Come on, come on. If I say superior ovary, that simply means if I see any flower, so the position of ovary should be on the higher side in comparison to the rest of the floral parts, which means your calyx, your corolla, your androsium, they are situated below the ovary. Then we can say this is my superior ovary. And if, let's say, we speak about inferior ovary, in that case, my ovary should be on the lower side. The position of ovary should be on the lower side. And my calyx, my corolla, my androsium should be on the position should be on the higher side. Then we say this is your inferior ovary. Okay, that is the difference first of all. Okay, if I speak about the symbols, So this symbol, this is for superior ovary, and this is for inferior ovary. Now please pay attention, kids. This hyphen represents position of rest of the floral parts. So if the hyphen is below the G, which means the rest of the floral parts are below in position with respect to ovary. If the hyphen is above the G, which means the rest of the floral parts are above than your gynosium. If they are above, then ovary is inferior. If they are below, then ovary is superior. As simple as that. Your question says, this is the symbol of inferior. No, this is the symbol of superior ovary. Okay? So the statement is incorrect, right? So answer is option number D. All right, everybody, with this, we have done 25 questions for this particular session. If you really like the session, then I'll really appreciate that you can hit the like button and share it with all your friends. And when the 2025 batch is already live, students, this is basically for NEET 2025 students. And this 1 year will definitely change your life. This 1 year will change the type of preparation that you are doing for NEET examination. Okay, so do check out the description box. There is a Google form attached to it. Then fill this Google form and help yourself. Yes, thank you very much for having me. We'll see you in the next classes. Do hit the like button, share it with all your friends, and subscribe to our very beautiful channel. We'll see you in the next class, same time, 3 to 4, in the next session. Bye, everybody. Till then, be happy, be healthy, take care of yourself. Bye, kids.